For this video, I will be spearheading on making the COVID-19 vaccine that is safe and effective enough to be approved by the FDA. First, I need to identify the antigen my vaccine would have. Instead of spending 10 to 15 years to develop a vaccine either containing an attenuated or amputated virus, I would choose a speed boost called mRNA technology that had been studied and used against coronaviruses by experts at NIH before the pandemic, shortening the process to less than a year. And so, I have made an mRNA vaccine, one with antigen being the mRNA that encodes for viral glycoproteins that are genetically identical to ones on the SARS-CoV-2 virus. Let's give it a name. So, what steps were taken to make mRNA 1273 and how does it work? And more importantly, is it effective and safe? Step 1. As mRNA is derived from DNA, I need to identify the gene on the DNA that encodes for the viral glycoprotein. Then I would produce that specific mRNA via in vitro transcription, where I would make a template of that gene and have RNA polymerase transcript that mRNA. As mRNA needs to be translated by a ribosome to synthesize the glycoprotein, we need to deliver it into our host cells. But because mRNA is negatively charged and will be repulsed by the anionic cell membrane, we need a delivery vehicle. Introducing lipid nanoparticles, a vesicle made of ionizable lipids that is positively charged at low pH and neutral at physiological pH, and other ingredients that help with binding to the cell membrane, such as this and this, and maintain the stability, being salt and sugar, of the mRNA. Once inside, they will release the mRNA. Upon reaching the ribosome, the mRNA will undergo a process called translation, where each codon of 3 bases on the mRNA signals for a specific anticodon, a tRNA, or transfer RNA that delivers specific amino acids, forming peptide bonds and eventually the glycoproteins. Once the viral glycoproteins are made, our cells will display them on their cell membrane. As soon as the immune system recognizes these viral proteins, antibodies are made to neutralize them and memory cells are made to acquire long-term immunity. So is it effective against future infection with SARS-CoV-2? Yes. Is it safe? For sure. Wait, a bonus power? Wow, the ability to make any viral vaccine faster than ever? With just an mRNA and LNP? That's a true victory.